Okay, this next video we are going to talk about RNA processing. This is a thing that happens to the RNA immediately after it's being created while it's still in the nucleus. So in the first part of transcription, the enzyme RNA polymerase made the brand new fresh piece of mRNA by copying the DNA and pairing up the nucleotides for the chunk that we wanted to copy, for the gene that we're interested in. And for our example, we're using hemoglobin. So the RNA polymerase II enzyme knows to start copying at the DNA at the promoter sequence, copies all the letters all the way along until it gets to the terminator sequence, and then it falls off. And so you get this piece of messenger RNA, which we call pre-mRNA, or sometimes you also hear it called primary mRNA. It's like it's in its first primary state, like it's brand new, fresh, nothing's been done to it yet. All right, so there are three things that you have to do to a piece of mRNA to turn it from pre-mRNA into what we call mature mRNA. And when it's mature, it means it's done and finished up and ready to leave the nucleus and go on to translation. So the three things are, okay, so number one, we have to protect the ends of the molecule. So RNA is much more fragile than DNA. DNA always has a partner. The two strands are always paired up and then they're wound up together in a helix, and that provides it with some degree of protection. And then the whole stuff is coiled as well. So DNA is pretty kind of sturdy. On the other hand, RNA is a single strand with just like exposed bases, and it has to go on this very long and treacherous trip out of the nucleus, through a little pore, into the cytoplasm where it has to bounce into everything and attach to a ribosome, and a bunch of things have to happen to it. So it's quite a long and perilous journey that this little RNA molecule has to take. And so we want to kind of protect it. And the first thing we do is protect the ends. I kind of think about this like protecting like a rope from fraying. You have to protect the ends, otherwise they kind of fray and the ends go bad. So you protect the two ends differently because they're chemically different, because one end is a 5' end, which has a phosphate group, and on the 5' end we add something called a cap. So that's the 5' cap. We stick that on one end of the primary mRNA. And then on the 3' end, we stick something called a poly-A tail. Now, a poly-A tail is basically, it means many A's, poly-A. So, you know, the RNA has a sequence. It might be G, A, U, A, U, G, G, C, C, G, C, U, A, whatever, blah, blah, blah. And then when it gets to the end of the actual message, the actual recipe, then it's just going to go A. We're just going to stick a whole bunch of nucleotides that say A on the end. This isn't recipe. This isn't anything significant. All this acts as is like a protection, like a bumper or a buffer at the end of the molecule. So if something accidentally happens and something runs into this and knocks off the end, you can knock off an A, and you can knock off an A, and you can knock off an A, but you haven't eaten into the important bit, which is the actual message back here. This is the important stuff. So the poly-A tail protects the 3' end, and then on the 5' end we put this thing called a cap. And it's basically just, it actually is like a hat, you know, like a hard hat to protect the end of it. But it's a special nucleotide kind of thing, like a modified nucleotide that can't be kind of eaten into very easily. It's not very chemically reactive. So that protects the 5' end. So RNA processing, 5' cap on one end, 3' tail on the other end, a poly-A tail on the other end. And now the last part is the most interesting, and that is we have to do something called splicing. And when we splice, what we do is we're taking out the introns. So let me write that down if I can spell it. Splicing. That looks like a kindergartner wrote that. I will try again. Splicing. All right, so why do we have to splice? Well, We didn't know about this until, I don't know, it wasn't that long ago, 20, 30 years ago, maybe even less, where somebody was studying the sequences of genes and the RNA that was copied from them. And they noticed that there was all kinds of junk sequence inside the genes. And this would be like reading a book or reading a poem that went, Mary had a blah, 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 little lamb, its blah, 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 fleece, blah, 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 was white as blah, 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 snow. Like all that blah, blah, blah got put in there. It shouldn't be there. It should just say, Mary had a little lamb, its fleece was white as snow. Right? It should just be that rhyme that everybody knows. But somehow all this extra words got thrown in there and made it not make sense anymore. And so the first time the researcher discovered this, they published it out there into the literature. And loads of scientists were like, no, you must have looked at that wrong. That can't be right. We know there's junk in the rest of the chromosomes, but the genes themselves, they can't have junk in them. The genes are really special. They have the recipes for proteins. If you mess them up, the proteins will be wrong. There can't possibly be junk inside the actual important sequence of the genes. Well, they did more and more work on it, and eventually, after lots and lots of sequencing and looking at all this stuff, people realized that actually, yes, it's true. Our genes have a lot of junk DNA that has somehow got into them. And there's reasons and explanations for how it's gotten in there. We kind of know a little bit more about this now. But anyway, for now, it's enough to understand that our recipes or our genes on our chromosomes have been disrupted over years and years of being copied and passed on from generation to generation. There's these parts in the genes that shouldn't be there. And those pieces of junk are called introns. All right, introns. And unfortunately, the RNA polymerase II enzyme that made this RNA can't tell introns. So the junk is the intron. The good stuff, that Mary had a little lamb, that's an exon. So the exon is the good stuff. The intron is the joke, the junk stuff. Unfortunately, all that the enzyme RNA polymerase II knows to do is start at the promoter and copy all the way to the terminator, everything, and just stop at the terminator. So it's going to copy everything, good stuff and junk. So before this piece of RNA leaves the nucleus to go out and get translated, you need to cut out all the junk. So it's a bit like editing a movie when you have all the movie scenes and you cut out the bloopers and stick back together the good stuff, and you end up with a whole movie of decent scenes without all the messed up stuff in between, right? And that's kind of where this idea of splicing comes from. That's why it's called splicing. That's what they used to do in the old days when movies were done on film. Nowadays, all digital. But so if we look at this diagram here, we can see we've put the cap and the tail on of this mRNA, but it's still got introns and exons in it. So the last step we have to do to finish RNA processing is cut out these introns. Get rid of this, and then glue this to this and this to this, so that we end up with a mature piece of mRNA that is just exon stuck to exon stuck to exon with all the introns removed. So it's kind of hard to remember this because it sounds like exon means exit, but it's the other way around. You cut out the intron. The intron is the stuff you want to take out. You want to leave the exons and glue them together to make your mature mRNA. That's what this picture is down here. And so once the pre-mRNA has had a cap on one end, a tail on the other end, and all the introns cut out, and it all stuck back together, now it's done and ready, and it's ready to leave the nucleus. It goes out through a little pore. There's lots of them, you can see right here. And once it goes out into the cytoplasm, it's going to go find a ribosome, and it will get translated, which is the next or the final step of gene expression.